Alright gang, here is the video for section 4, the final section of chapter 11 on cell communication. And now we finally get to the response. And cell communication, we've received a signal and sent it through the cell transduction, and now we get to the response. And how do cells actually respond to the signals that they've received. So as you read through the chapter, or as you're going to read through the chapter, the book referenced epinephrine a bunch of times, and it actually went through the whole pathway for epinephrine. So I'm going to use that as the first example of a type of response, cellular response to activation or to communication. First of all, epinephrine, the other name that it commonly goes by is adrenaline, and so essentially, whenever you get a rush of adrenaline or a rush of epinephrine, it's going to do a couple things. One, it's going to try to increase the amount of sugar that's in your blood so that you have energy to do something with whatever caused you to have an adrenaline rush. So that's one thing it's going to do, and then there's actually a whole bunch of things. It just depends what cells it gets into. The cells in the liver are the ones that are going to try to increase the amount of sugar that goes in your bloodstream. And just kind of a background on this, when you eat a lot of sugar, your body stores a whole bunch of it as glycogen in your liver. Okay, and that's one of the liver's jobs. So it stores it till later. So you've got the store of glycogen, which is basically stored sugar, and then epinephrine is going to end up triggering that sugar to be taken out of storage so that it can go into the so you can get energy from it. So anyway, I'm actually going to step through everything here from the time that you receive the signal, and kind of the goal on this with your response to cell-to-cell communication. There's going to be either a response that happens in the cytoplasm of the cell, or there's a response that could happen in the nucleus of the cell. Epinephrine happens to be one that's going to make a response in the cytoplasm. Okay, so if we go all the way to the beginning, step one in cell communication is going to be the reception of a signal. And so this particular epinephrine, this pathway, works through G protein linked receptor. So we've got epinephrine up here, and it is going into the active site, basically, or the receptor, the G protein linked receptor. If you remember how these G protein linked receptors work, there's an inactive G protein that's going to go over to the protein receptor, the G protein receptor. If epinephrine happens to be in the receptor, then our G protein is going to go from having GDP to having GTP, which activates it. So we have our activated G protein here. It's going to go over to an enzyme, adenylyl cyclase, which we learned about before, but it's going to go over to that and activate that enzyme. And then that enzyme that is now activated is going to turn ATP, remove two of the phosphates, and change the shape a little bit, and it's going to become cyclic AMP. If you remember, cyclic AMP becomes a second molecule, a second messenger molecule, that's going to relay the message to a protein kinase molecule, and it's going to activate a protein kinase molecule, and now we have the beginning of transduction. So I'm going to move myself up here, and you can see our transduction pathway as we saw it in the last video, and it's also over on the right here, we're seeing the the abbreviation for the diagram, but anyway, so we have our activated protein kinase A here, and it's going to phosphorylate another protein kinase, and that's going to phosphorylate another protein kinase, and here we get to the payoff. That final protein kinase is actually going to phosphorylate something called glycogen phosphorylase. Okay. Glycogen phosphorylase, once it's activated, here we get our cell response. It has the ability to go to glycogen, that's its substrate, and it can take glycogen, turn it into glucose 1-phosphate. Glucose 1-phosphate is one of the intermediates in glycolysis. So basically it's sugar, a form of sugar, but that can be released into the blood to get to the cells that need energy, and they can jump right into glycolysis. Okay. This all happens in the cytoplasm, as it's shown in these diagrams, a whole bunch of steps, but here we have from reception to response, the whole big long pathway where we're signal, being sent to signal, being sent to signal, or whatever, and then ultimately it's doing the work all of this and the work that has to be done is getting sugar out of storage so it can be released. Okay, so that's all the steps of epinephrine. Not a hugely complex situation here, but it is a lot of steps, and we're going to talk about a little bit later on why for the lot of steps. Now we're going to look at a scenario where we have the response happening in the nucleus rather than in the cytoplasm. One thing I'll say before we move on is these cytoplasm responses tend to be something metabolic, like changing one molecule into another. In this case, it's changing glycogen into a bunch of glucose 1-phosphates. It's a metabolic response. We'll see the ones in the nucleus end up typically being like a genetic response, is what we're going to look at there. So next slide. All right, so here we have the response as it would be happening if it was in a nucleus, if the response is going to be in the nucleus. And if you remember earlier, one of the videos, me talking about growth factors, and growth factors being one example of a growth factor being if you were to get a cut or something like that, that cells near the cut will release growth factors that tell other cells that are in the area, start dividing, we need to do some sort of healing here. That's one type of growth factor. There's all kinds of growth factors. But anyway, this is again showing all three of the steps in our response here, but this one's going to culminate in the nucleus rather than in the cytoplasm. So we have, they just show this reception step up here where the growth factor attaches to some sort of a receptor. It doesn't say which type, and my guess would be all the different types of growth factors, probably some of them use the G protein as a way of reception. Some of them probably use the tyrosine kinases. It just depends. But anyway, somehow it is being received, and the signal is getting to the inside of the cell, and then we're having a cascade, phosphorylation cascade, which would be a bunch of protein kinases, just like we saw before. But in this one, the last protein kinase that gets activated is going to enter the nucleus of the cell. When it gets in there, it is going to activate a transcription factor, and sometimes it'll join something, and the two of them combined will become a transcription factor. But either way, it is going to, these transcription factors basically turn on genes. And so in the case of a growth factor, that typically means we've got a cell that's not dividing, and the growth factor is going to tell it, you need to prepare to divide. And so the growth factor, the signal when it gets here, the genes that it's turning on is one of the genes that says we're going to divide, we're going to make more cells, and that's a big whole deal that happens there. But this one culminates in turning on and off genes in the nucleus, and the growth factor is a good example of that. And my understanding always was that the majority of signals received by a cell are going to end up being a nuclear response rather than a cytoplasmic response. I don't know what the numbers are on that. So, all right, we'll go to the next slide here. Okay, this slide has to do with a question I had asked in the last video, but the question being, why are there so many stinking steps in these things? Why can't it just be a molecule enters a cell and activates what it's supposed to activate? Like, why does it have to be a phosphorylation cascade and all these intermediates and activating things and then deactivating them and all of that? And there's a couple benefits to it. So when you ask, like, what's the reasoning behind something happening, that gets kind of hairy in biology. But anyway, if you say what are the benefits of, so one of the benefits that scientists find, I'm going to slide myself over here, one of the benefits that scientists find in having a lot of steps, one is it allows the signal to be amplified, they call it. And anytime you amplify something, it just means you're making it more impactful. If you amplify sound, you take a small sound, and when you amplify it, it becomes really loud. Well, in this case, we can take one, and this goes back to the epinephrine one over here. They have written down numbers of molecules, quantity of molecules. And if I look at the epinephrine, one molecule can hit a cell, just one molecule, and it's going to activate a G protein, but it can actually activate several hundred G proteins just from that one molecule landing on the receptor. So you get to 10 to the second molecules, which is about, it's several hundred molecules being activated of the G protein. Each of those G proteins can then go and activate the adenylyl cyclase. Okay, so we're at several hundred of those. But then when you start the adenylyl cyclase that's activated, it can take a lot of ATP and turn it into cyclic AMP. And so when we do that, all of a sudden we get to 10 to the fourth, which gets to tens of thousands of molecules of cyclic AMP, which are going to then activate tens of thousands of molecules of protein kinase A. But then that protein kinase A can activate several molecules of the next protein kinase. So then you get 10 to the fifth, you're getting the hundreds of thousands of molecules that are activated, go to the next protein kinase, or the next step here, the glycogen phosphorylase, and you get up into the millions of glycogen phosphorylases that are activated, which means we had one epinephrine molecule land on this cell, and we're going to end up having millions of glycogen molecules turned into glucose 1-phosphate, and that can happen a lot of times. You get to 10 to the eighth. That's 100 million molecules of glucose 1-phosphate being made from this one epinephrine. So this becomes pretty powerful, this amplification idea. And so that's one of the advantages of having a bunch of steps, is that instead of having one molecule making one molecule of epinephrine resulting in one glucose 1-phosphate being produced, it's ending up getting hundreds of millions of the product that we want. Okay, so that's a really good thing, really powerful thing. So there's that amplification idea. The next idea here is that by having a lot of steps, it actually increases the diversity of responses that you could get from one signal, and by having a diversity of ways that it could be interpreted, it ends up meaning that any cell, specific cells within the body, can all have their own specific response to a signal. So what this is showing is 4 different responses to the same signal. So actually the first and second and third, they're all using the same receptor protein, but then we have the fourth situation over here, sorry, 1, 2, 3, 4, response 5, sorry, is a different receptor protein. Okay, and so there's a cell that would have a specific situation there. We can also look at, if I compare 1 and 2, and these would be different cells, but they've got the same initial receptor, and they're just resulting in different sets of molecules going down through them. Anyway, I'm not going to go into the detail on all of this, but it gives us more options of how a signal protein or a signal molecule can be interpreted by cells, and that's how you get something like the liver cells respond to epinephrine by making sugar available to the blood, but the heart cells increase how often they pulse. Okay, so your heart rate increases and becomes more powerful actually when epinephrine is released. So you get 2 different responses because we have all these steps. If it was something as simple as we've got a signal molecule, it enters a cell and it does its one thing, there'd be a limited number of responses we could have for the one molecule that it's talking about. So that's some of the answers to why there's so many steps. It just gives us some advantages along the way. Okay, quick slide here that goes through how could we make this whole thing a little more efficient. The reasoning for this question here is, if we've got these proteins in the cytoplasm and everything is relatively random that happens in a cytoplasm, so technically you would have all these relay molecules that have to randomly bump into each other, and if they are on total different sides of the cell, then our response could be pretty slow if molecules have to get from one side of the cell to the other side of the cell. So one way to make things a little bit more efficient is to say, let's hold all of the proteins that are in a transduction pathway, let's hold them all together. And there's 2 ways we can do that. This diagram shows a scaffolding technique where you have one large protein that has a bunch of smaller proteins attached to it. And so this one, there's the scaffold protein. It has 3 different protein kinases. And so you literally have the product of one protein kinase right beside the next one. It can act on it immediately, which is right beside the next one, which can act on it immediately. And it can just speed up the whole process. So that's a scaffolding idea. There's another idea that's very similar, but it's complexes, where you just take a bunch of proteins and attach them together in one large complex. The difference is that this one has the diagram shown with the scaffolding has one large protein molecule with other ones attached, whereas if you just have a complex, it's more of just having a bunch that are attached to each other, not necessarily a main one with several on it. So that's the one way we can make things more efficient. The next big question is, how do you turn a signal off? If you want cell like growth factors, if you want there to be growth factors stimulating growth now, what do you do when you want it to stop? How do these things stop? And that's the next slide. So the way we turn signals off is basically by deactivating different molecules. And so I'm just going to go through 3 different situations that the book outlines with deactivating molecules that were active. So if we look at our G proteins, they get activated when there's GTP on them, and essentially the G protein itself ends up being responsible for hydrolyzing the GTP, which just means doing a hydrolysis reaction and chopping off the terminal phosphate on the GTP, turning it back to GDP, and then it just deactivated itself. So if there's a G protein that is causing a signal pathway to get started, it'll actually inactivate itself, which very quickly turns off the signal. If we look at a different concept here, this is going with another G protein, but we're taking it a step further along when we look at the cyclic AMP as being that second messenger oftentimes. And so we have cyclic AMP here, and if it's going to be deactivated, needs to be turned into something other than cyclic AMP. And so there are enzymes called phosphodieesterases. Anyway, the bond between the first phosphate and that one phosphate in the AMP, if we can add 2 more phosphates to that, if we can break a bond within there, it's called a phosphodieester bond. But anyway, then it'll get rid of the cyclic part of cyclic AMP and just make it AMP. Very, very similar structurally, but it does not do its job anymore. And that's a way that cells deactivate that cyclic AMP, and then then that'll end whatever it's activating. So that's that one. And then the final one, and this video is getting a little long here, so would be with the phosphorylated protein kinases. And there are enzymes floating throughout the cell called phosphatases, and they basically chop phosphates off of protein kinases. And so the cell is producing enzymes that are going to turn off these molecular pathways here, so that they don't continue, and this will happen naturally. Those phosphatases are constantly inside of a cell, and so if we have activated protein kinases, they're going to get deactivated, and our signal is going to stop. If we want them all to get reactivated, then we just need more signal molecules back at the beginning, and we can reactivate, they'll get deactivated again, and so on. So that wraps up our chapter 11. We're done. Hopefully you guys can come up with some questions to ask either online or in class, and hopefully these videos have been helpful.